Hey, science fans! Get ready to embark on a fascinating journey into the microscopic world of cells. Ever wonder how you grow from a tiny dot, a single fertilized egg, to a full-sized you, a complex organism with trillions of cells? It's a mind-boggling transformation, isn't it? Or consider this. How does your body heal a paper cut, seemingly knitting itself back together? What invisible processes are at play? It's all thanks to the cell cycle. This fundamental process is the unsung hero behind growth, repair, and even the simple act of staying alive. The cell cycle is a precisely orchestrated series of events that lead to a cell's growth, DNA replication, and ultimately, division. It's a carefully choreographed dance of molecules and structures. This division results in the creation of two new daughter cells, each carrying a complete set of genetic information, ready to start their own cycles. It's like a cell's life story, a continuous loop of growth, replication, and division. Think of it as a journey from birth to growing up, maturing, and finally, contributing to the next generation by making new cells. Growth, the increase in size and complexity. Healing, the remarkable ability to mend injuries and restore damaged tissues, and replacing old, worn-out cells with fresh new ones. All these vital processes depend on this intricate cycle. The cell cycle is happening in you right now. Every second, millions of cells are dividing and replicating within your body. It's the engine of life, a fundamental process that keeps us alive and functioning. If it stops, slows down, speeds up, or goes wrong in any way, big problems can happen, potentially leading to diseases like cancer. So understanding it is key to understanding life itself and to developing new treatments for diseases. Ready to dive deeper into the fascinating world of the cell cycle? Let's go. We'll explore the different phases, the key players, and the critical checkpoints that ensure everything runs smoothly. The cell cycle, a fundamental process of life, has two main parts, and the first is interphase. Think of it as the cell's big prep party. It's a crucial period where the cell gears up for the dramatic event of cell division. During interphase, the cell grows significantly, increasing in size and mass. It's like a plant seedling sprouting and reaching for the sun. It also diligently does its job, carrying out its specific functions within the organism, whether it's a nerve cell transmitting signals or a muscle cell contracting. And most importantly, it gets ready to divide, meticulously preparing all the necessary components and machinery for the upcoming cellular split. Interphase isn't just one long stretch, it has three distinct phases, each with its own set of tasks and checkpoints. First, there's G1, then S, and finally, G2. In G1, the cell grows, increasing in size and accumulating the necessary resources. It actively makes proteins and organelles, the tiny machines that perform specific functions within the cell. In S phase, the most critical event occurs. DNA is copied. Each chromosome meticulously makes an exact copy of itself, ensuring that each daughter cell receives a complete set of genetic instructions. These identical copies then pair up, forming what we call sister chromatids. G2 is the final check. The cell grows a bit more, ensuring it has enough size and resources. It also preps for division, making final adjustments and gathering all the necessary tools. The cell rigorously checks if everything's ready, DNA copied without errors, and essential supplies are fully stocked. It's like a pilot running through a pre-flight checklist. If all systems are go and the cell meets all the criteria, it confidently moves to the next stage, M phase, the exciting stage of actual cell division. Let's see what happens next as the cell embarks on this incredible journey of splitting into two. How does a cell make sure everything goes right? Checkpoints. These are like traffic lights, making sure the cell is ready before moving on. The G1 checkpoint checks size, nutrients, and DNA damage. If something's wrong, the cell pauses or fixes it. The G2 checkpoint ensures DNA was copied correctly, and the cell is ready for division. If there's a problem, the cycle stops. This helps prevent diseases like cancer. During mitosis, the M checkpoint checks if chromosomes are attached to spindle fibers. If not, division halts to prevent errors. These checkpoints are the cell's quality control. Interphase is done. The cell has grown, replicated its DNA, and is now fully prepared to divide. It's like a runner stretching and getting ready at the starting line, poised to begin the race. All systems are go. Now comes M phase, or mitosis. This is the main event, the actual division process where one cell becomes two. It's a carefully choreographed dance of chromosomes and cellular machinery. Mitosis divides the nucleus, that control center of the cell. Think of it as carefully dismantling the headquarters and preparing to duplicate it perfectly. The nucleus, with all its genetic information, needs to be split evenly. Then the cell splits, making two identical daughter cells. It's like photocopying the original cell, creating a perfect replica. Each daughter cell now has its own fully functional nucleus and is ready to carry out its specific role. Mitosis has 4 stages, each with its own distinct set of events. These stages ensure that the chromosomes are properly sorted and distributed. It's a complex process, but it can be broken down into manageable steps. Prophase, the first stage; metaphase, the second stage; anaphase, the third stage; and telophase, the final stage of mitosis. Remember PMAT, a handy acronym to help you recall the order of the stages: prophase, metaphase, anaphase, and telophase. Keep this in mind as we delve deeper into each phase. In prophase, chromatin condenses into visible chromosomes. The long, tangled strands of DNA begin to coil tightly, becoming more compact and easier to manage during the division process. The nuclear envelope breaks down, and spindle fibers form. The protective barrier around the nucleus disappears, allowing the spindle fibers to access and organize the chromosomes. In metaphase, chromosomes line up in the middle, forming what's called the metaphase plate. This alignment is crucial for ensuring that each daughter cell receives an equal and complete set of chromosomes, attached to spindle fibers. These fibers act like tiny ropes, connecting to the chromosomes and preparing to pull them apart. Anaphase, the separation stage. Sister chromatids separate, pulled apart by the spindle fibers, and are pulled to opposite ends of the cell. The spindle fibers shorten, drawing the sister chromatids towards the poles of the cell. The cell elongates as chromatids move apart. This stretching helps to physically separate the two sets of chromosomes, ensuring they don't get tangled up. Each chromatid is now a chromosome, a complete and independent unit of genetic information. This process ensures each new cell gets a complete set of DNA, guaranteeing that they can function properly and carry out their designated tasks. Let's finish the split. With the chromosomes safely at opposite ends, the cell can now divide completely, giving rise to 2 new, identical cells. After anaphase comes telophase T for 2 nuclei. Chromosomes reach the poles uncoil and new nuclear envelopes form. The cell now has 2 nuclei and spindle fibers disappear. Cytokinesis divides the cytoplasm, pinching the cell in 2. 2 identical daughter cells are formed. Voila! Mitosis is how you grow, heal, and replace old cells. Without it, multicellular life wouldn't exist. Mitosis is the hero behind growth and repair. Now let's talk about making babies. Sexual reproduction needs a different process. Meiosis. Meiosis makes gametes, sperm and egg cells with half the usual chromosomes. It has 2 rounds of division: meiosis 1 and 2. Starts with 1 diploid cell, ends with 4 haploid cells. In meiosis 1, homologous chromosomes pair up and exchange DNA. Crossing over creates genetic variety. Homologous chromosomes then separate. Meiosis 2 is like mitosis. Sister chromatids separate. Result: 4 unique haploid cells, each genetically different. This variety is crucial for sexual reproduction. Meiosis shuffles the genetic deck. Let's compare mitosis and meiosis. Mitosis, 1 division, 2 identical diploid cells, used for growth, repair, and asexual reproduction. Happens in body cells. Meiosis, 2 divisions, 4 unique haploid cells, used for making gametes. Happens in reproductive organs. Mitosis is like a photocopier, exact copies. Meiosis is like shuffling cards, new combinations. Genetic diversity from meiosis drives evolution and survival. The cell cycle is the foundation of life, growth, repair, and new generations. Science is everywhere, and it's awesome.